Hello there! Welcome back! Before we jump in, if you haven't watched part 1 on the plasma membrane yet, stop right here. Because everything we're about to cover builds on it. The structure of the plasma membrane, the lipid bilayer, the membrane proteins, the concept of selective permeability. That's the foundation. Without it, what follows won't fully click. All good? Great! Let's get into it. In this video, we're going deeper. We're going to explore exactly how drugs cross biological membranes. Here's the big picture. Drug absorption across membranes determines bioavailability, meaning how much actually reaches your bloodstream. But here's the catch. Your body doesn't let everything in. Epithelial barriers, like your intestinal lining, act like strict gatekeepers, deciding what passes and what gets blocked. At the center of it all is the plasma membrane, a smart, semi-permeable barrier, and only drugs with the right size, polarity, and solubility make it through. So how do drugs actually get across? There are two main pathways. First, the transcellular route. The drug passes straight through the cells. Second, the paracellular route. Instead of going through the cells, the drug squeezes between them, through tiny gaps called tight junctions. These two routes are completely different in how they work, how permeable they are, and what types of drugs use them. Understanding these pathways is essential. It's what lets us predict how a drug gets absorbed and how it reaches its target. So let's break them down, starting with the transcellular route, its mechanisms, real-world drug examples, and why it matters clinically. All right, let's zoom in on the transcellular route. The transcellular route is exactly what it sounds like. The drug moves through the epithelial cells themselves. To do that, it has to cross two membranes: the apical membrane, the side facing the lumen, and the basolateral membrane, the side facing the bloodstream. It's a journey through the cell, not around it. This is the major pathway for drug absorption, especially for lipid-soluble or lipophilic drugs. And here's why that makes sense. If the membrane is made of lipids, lipid-loving molecules are going to slip right through. Let's walk through it step by step. Step 1. The drug diffuses through the apical membrane, entering the cell from the intestinal lumen or surrounding extracellular space. Step 2. It travels through the cytoplasm, crossing the cell's interior. Step 3. It diffuses across the basolateral membrane and exits toward the blood. Step 4. It enters systemic circulation. Now it's in the bloodstream and ready to do its job. Remember, the membrane is a lipid bilayer. So lipid-soluble, non-ionized drugs glide through easily. Polar or charged molecules, they struggle. That distinction is going to matter a lot as we go on. Now let's talk about the different types of transcellular transport. The first one is the engine behind most of this: simple passive diffusion. This is the primary way most drugs cross cell membranes to reach your bloodstream. Simple or passive diffusion is the movement of molecules from an area of higher concentration to an area of lower concentration, downhill, essentially. No energy needed, no carrier proteins. The molecules just flow naturally until both sides of the membrane reach equilibrium. It's physics doing the work. The principle is beautifully simple. Molecules flow along their concentration gradient. For a drug sitting in your intestine, that means moving from the lumen, through the epithelial cells, and into the blood. The bigger the concentration difference, the stronger the driving force. So what makes passive diffusion unique? Here are its key characteristics. No energy required. The cell doesn't need to burn any ATP for this to happen. No carrier proteins. Molecules move on their own. No assistance needed. Non-saturable. Because there are no transporters to overwhelm, the process doesn't max out, even at high drug concentrations. Non-specific. It doesn't discriminate. Any molecule with the right physical and chemical properties can diffuse through. And concentration gradient dependent. The steeper the gradient, the faster the diffusion. Simple as that. Now, passive diffusion follows a precise mathematical rule. Fick's law of diffusion. Fick's first law tells us exactly how fast diffusion happens. The rate of diffusion, J, equals membrane permeability, P, times surface area, A, times the concentration difference across the membrane, C1 minus C2. The negative sign just confirms that diffusion always moves from high to low concentration, always downhill. Put simply, diffusion speeds up when the concentration gradient is steeper, the surface area is larger, or the membrane is more permeable. Tweak any one of these, and you change how fast the drug is absorbed. Now let's trace the actual journey of a drug through passive diffusion. 4 steps. Step 1. Dissolution. The drug must dissolve in the surrounding biological fluids, like GI fluids. A drug that can't dissolve can't diffuse. It's that simple. Step 2. Partitioning into the lipid membrane. The drug has to dissolve into the lipid bilayer itself. Lipophilic drugs do this easily. Hydrophilic drugs, not so much. Classic examples here are diazepam and propranolol. Step 3. Diffusion through the bilayer. The molecule moves through the hydrophobic core of the membrane. How fast? That depends on lipid solubility, molecular size, and degree of ionization. Step 4. Exit. The drug emerges on the other side, enters the cytoplasm or extracellular fluid, and eventually reaches the bloodstream, where it can finally do what it was designed to do. What actually controls how fast passive diffusion happens? Let's go through the key factors. Lipid solubility. The more lipophilic a drug is, the faster it diffuses. We measure this using the partition coefficient. Molecular size. Smaller molecules slip through more easily. Larger ones have a harder time. Degree of ionization. This is a big one. Only non-ionized molecules are lipid-soluble enough to cross. Once a drug becomes ionized, it gets trapped on one side of the membrane. So what determines whether a drug is ionized or not? 2 things. The pKa of the drug, a measure of how easily it gives up or accepts a proton. The environmental pH, the acidity or basicity of the surrounding fluid. This is captured mathematically by the Henderson-Hasselbalch equation. Don't let the name intimidate you, it's actually quite logical. For weak acids, pH equals pKa plus log of A minus divided by HA. For weak bases, pH equals pKa plus log of B divided by BH plus, where a minus and BH plus are ionized forms and B and HA are non-ionized forms. Surface area. This is why the small intestine is so good at absorption. All those villi and microvilli dramatically increase the available surface area, and more surface area means faster absorption. Membrane thickness. The thinner the membrane, the shorter the distance the drug has to travel, and the faster it diffuses. And blood flow. Good circulation continuously sweeps drug molecules away from the membrane on the blood side, keeping the concentration gradient steep. No flow, no gradient. No gradient, no diffusion. Now, passive diffusion doesn't always happen the same way. There are actually 2 forms of it. Form 1. Lipid diffusion. The drug dissolves in the lipid membrane and diffuses across it. This is the most common mechanism of drug absorption. Examples: aspirin and propranolol. Form 2. Aqueous diffusion. Some very small hydrophilic molecules can pass through aqueous pores or channels in the membrane. Think of them as tiny water-filled tunnels. This pathway is less common. Examples include small ions and water molecules. Why does all of this matter clinically? Passive diffusion is important because it allows rapid drug absorption. It does not require cellular energy. It occurs in many tissues throughout the body. But it's not perfect. Passive diffusion has real limitations. Large molecules cannot pass easily. Highly polar or ionized drugs diffuse poorly. The process depends heavily on the drug's physicochemical properties. Now let's move to something a little more sophisticated. Facilitated diffusion. Here's a question worth sitting with. Not all molecules can pass through the lipid membrane on their own. So, how do polar molecules, ions, and certain drugs get in? The answer is facilitated diffusion, a specialized carrier-mediated transport mechanism that gives those molecules a helping hand. Facilitated diffusion is still movement from high to low concentration, still downhill. But now the molecule gets help from specific transport proteins, and crucially, it still doesn't require cellular energy. Why do we even need this? Lipid-soluble drugs can slip right through the membrane. We've covered that. But many important molecules are polar or charged. They simply cannot dissolve in the lipid bilayer. They need a different route. That's where protein pathways come in. How does it work? Facilitated diffusion works through two types of membrane proteins: carrier proteins and channel proteins. Both provide a hydrophilic pathway, allowing molecules to bypass the hydrophobic membrane core. Carrier proteins bind the molecule and physically transport it across the membrane. These proteins undergo reversible conformational changes during transport. Example: amino acid transporters. Here's where it gets really clever. Some drugs are designed to mimic natural molecules so they can hijack these transport systems. Examples include levodopa, transported by amino acid transporters; gabapentin, transported by L-amino acid transport systems; methyldopa, transported by amino acid carriers. These drugs resemble amino acids and use amino acid transporters to cross membranes. Channel proteins. Channel proteins form hydrophilic pores in the membrane, allowing ions or small molecules to pass through. These include ion channels and water channels called aquaporins. However, most drug transport through facilitated diffusion involves carrier proteins rather than channels. Steps in facilitated diffusion. Step 1. Binding. The molecule finds its match. The molecule binds to a specific carrier protein on the membrane, and these proteins are highly selective. They recognize molecules based on their shape and chemical structure. Amino acid transporters, for instance, recognize molecules with a similar structure to amino acids. A drug like levodopa can use this system because it resembles natural amino acids. Step 2. Conformational change. The protein undergoes a shift. Once the molecule is bound, the carrier protein physically changes shape. A conformational change. This shift flips the molecule from one side of the membrane to the other. The molecule is carried across, never touching the lipid bilayer. Step 3. Release. Mission accomplished. Once the carrier protein reaches the opposite side of the membrane, the molecule is released into the cytoplasm or extracellular fluid. The carrier protein then returns to its original conformation, ready to transport another molecule. Here's what sets facilitated diffusion apart. Carrier-mediated. Requires specific transport proteins. No energy required. Molecules move along the concentration gradient. Saturable. A limited number of carriers means the transport process can become saturated. Specificity. Carrier proteins are highly selective, meaning they transport only specific molecules or molecules with similar structures. And competition. Molecules with similar structures may compete for the same transport protein, which can reduce the rate of transport for each molecule. Why does facilitated diffusion matter? Because it helps transport essential molecules like glucose, amino acids, and ions into cells, things your body simply can't function without. It plays a crucial role in maintaining normal cellular activity and energy supply. Even more importantly, it helps move substances across specialized barriers like the blood-brain barrier. By understanding these transport systems, scientists can design drugs that mimic natural molecules, helping them enter cells more effectively. It also helps improve drug absorption and predict drug interactions when multiple drugs compete for the same transporter. So this isn't just theory. It's the foundation of smarter drug design. And in pharmacology, it allows certain drugs to enter cells by using these transport systems. Simple process, but absolutely vital for life. Now we're stepping it up. Let's talk about active transport, where the cell actually expends energy to move drugs against the current. Here's the definition: the movement of molecules across a biological membrane from a region of lower concentration to a region of higher concentration using specific carrier proteins and metabolic energy. usually ATP. Think about it. Moving uphill requires effort, and that's exactly what active transport does. It moves molecules against their concentration gradient, which means energy must be spent. The mechanism: membrane-bound transport proteins that act as molecular pumps, physically pushing molecules to where they need to go. Let's walk through the steps. Step 1. Drug binding to the transport protein. The drug or molecule first binds to a specific carrier protein located on the membrane surface. These carrier proteins are highly selective and recognize molecules based on their chemical structure. Many drugs resemble natural substrates such as amino acids, peptides, or organic ions, which allows them to use these transport systems. Examples include methotrexate and penicillin. Step 2. Energy supply. Energy for active transport is usually obtained from ATP hydrolysis. ATP provides the energy required for the transport protein to change its shape and move the molecule across the membrane. Some transport systems also use ion gradients, such as sodium gradients, as an indirect energy source. Step 3. Conformational change of the carrier protein. After binding the molecule and receiving energy, the transport protein undergoes a conformational change. This structural change allows the molecule to move from one side of the membrane to the other. Step 4. Release of the molecule. The molecule is released on the opposite side of the membrane, often in an area where its concentration is already higher. The carrier protein then returns to its original configuration and is ready to transport another molecule. Now, active transport isn't one size fits all. It comes in two main forms. Form 1. Primary active transport. In primary active transport, energy comes directly from ATP hydrolysis. The cell breaks down ATP to power the pump. The transport protein acts as an ATP-driven pump. A classic example is the sodium-potassium pump, which maintains ionic balance in cells. Although this pump mainly transports ions, similar ATP-driven pumps can transport drugs and metabolites. Form 2. Secondary active transport. Secondary active transport is more indirect. Instead of using ATP directly, it piggybacks on ion gradients, usually the sodium gradient set up by primary active transport in the first place. Two types of secondary transport exist: symport, co-transport. Both the drug and the ion move in the same direction across the membrane. Example: sodium-glucose co-transport in the intestine. Antiport, exchange transport. The drug and ion move in opposite directions across the membrane. Example: organic ion transport systems in the kidney. Let's lock in the key properties of active transport. 1. Energy requirement. Active transport requires metabolic energy, usually in the form of ATP. 2. Carrier-mediated. The process depends on specific membrane transport proteins. Saturation kinetics. Because the number of carrier proteins is limited, the process is saturable. At high drug concentrations, all transporters may become occupied, resulting in a maximum transport rate. Specificity. Transporters recognize molecules with specific chemical structures, often resembling endogenous compounds. Competition. Two molecules with similar structures may compete for the same transporter, potentially affecting drug absorption or elimination. Directionality. Active transport can move substances against their concentration gradient, whereas passive diffusion cannot. Where in the body does active transport happen? Active transport plays an important role in several organs. Intestine. Certain drugs use transport proteins to enhance absorption. Example: levodopa utilizes amino acid transporters for intestinal absorption. Kidney. Active transport in renal tubules is responsible for drug secretion and reabsorption. Examples include penicillin and methotrexate. Liver. Hepatocytes use active transport systems to move drugs into bile for elimination. Example: rifampicin, actively transported into bile. Blood-brain barrier. Active transporters regulate drug entry into and removal from the brain. Example: levodopa, actively transported into the brain via an amino acid transporter. So why does all of this matter? Here's the clinical significance. Drug absorption. Some drugs rely on transport proteins for efficient intestinal absorption. Drug interactions. Two drugs using the same transporter may compete, reducing the absorption or elimination of one of them. Drug resistance. Efflux transporters can pump drugs out of cells, reducing drug effectiveness. And drug elimination. Active transport in the kidney and liver helps remove drugs from the body. And finally, vesicular transport. Some molecules are simply too large, too polar, or too complex for any of the mechanisms we've discussed. They can't diffuse across. They can't use carriers. So what does the cell do? It gets creative. It wraps them up. Vesicular transport works by forming membrane-bound vesicles. Essentially, the cell membrane folds around a substance and creates a little bubble to carry it inside. When substances are brought into the cell, the process is called endocytosis. This mechanism is particularly important for the uptake of large molecules, such as proteins, peptides, nanoparticles, and macromolecular drugs. Endocytosis is defined as the process by which cells engulf extracellular substances is by forming membrane-bound vesicles that transport the material into the cell. Vesicular transport occurs through several steps. Recognition and binding. The substance to be transported first comes into contact with the cell membrane. Sometimes this interaction occurs non-specifically, while in other cases, the substance binds to specific receptors on the cell surface. 2. Membrane invagination. After binding, the cell membrane begins to fold inward around the substance. This folding forms a pocket-like structure that gradually encloses the material. 3. Vesicle formation. The membrane pocket continues to deepen and eventually pinches off from the membrane, forming a vesicle containing the substance. This vesicle moves into the cell's cytoplasm. 4. Vesicle processing. Once inside the cell, the vesicle may fuse with lysosomes for degradation, release its contents into the cytoplasm, and transport the substance to another part of the cell. Types of endocytosis. Endocytosis occurs in several forms depending on the type of material being transported. Type 1. Phagocytosis. Cell eating. You may have heard this one before. In this process, the cell engulfs large solid particles, such as bacteria or cellular debris. Specialized immune cells, such as macrophages and neutrophils, perform phagocytosis. Steps in phagocytosis. Recognition of the particle. 2. Extension of pseudopodia around the particle. 3. Formation of a phagosome. And 4. Fusion with lysosomes to digest the particle. Phagocytosis is essential for immune defense, removal of dead cells, and elimination of pathogens. Type 2. Pinocytosis. Cell drinking. If phagocytosis is cell eating, pinocytosis is cell drinking. In this process, the cell takes in small droplets of extracellular fluid along with dissolved substances. Unlike phagocytosis, pinocytosis occurs in almost all cells. Characteristics are non-specific uptake, continuous process, and small vesicles formed. Pinocytosis allows cells to sample extracellular fluid and absorb dissolved molecules. Type 3. Receptor-mediated endocytosis, the most selective. This is the most targeted and efficient form of endocytosis, and the most pharmacologically relevant. Specific molecules bind to receptors on the cell membrane, triggering vesicle formation. These vesicles often form in specialized membrane regions called clathrin-coated pits. Mechanism include ligand binds to receptor. 2. Receptors cluster in coated pits. Membrane invaginates. And 4. Vesicle forms and enters cytoplasm. Example cholesterol uptake occurs through receptor-mediated endocytosis of low-density lipoprotein, LDL particles. Endocytosis and the future of drug delivery. This is where pharmacology meets cutting-edge medicine. Endocytosis is at the heart of some of the most advanced drug delivery systems being developed today. Nanoparticles, drug-loaded nanoparticles, can enter cells through endocytosis. Liposomes. Liposomes can fuse with the cell membrane or enter the cell via vesicular transport. And antibody-based drugs, therapeutic antibodies, bind to receptors and enter cells via receptor-mediated endocytosis. The next type of vesicular transport is exocytosis. Exocytosis is the fundamental cellular process by which cells move large molecules, such as proteins, neurotransmitters, or waste products, from the interior of the cell to the extracellular space. This is achieved through the use of membrane-bound vesicles. The mechanism is a highly coordinated series of steps involving the cell's shipping and receiving department. Step 1. Vesicle trafficking. The Golgi apparatus packages molecules into vesicles. These vesicles are transported along the cytoskeleton to the plasma membrane. Step 2. Tethering and docking. Vesicles make contact with the membrane. Proteins called snares pull the vesicle close to the membrane. Step 3. Fusion and release. The vesicle membrane fuses with the cell membrane. The contents are released into the extracellular space, and the vesicle membrane becomes part of the cell membrane. Your cells have two ways to ship out molecules: the constitutive path and the regulated path. Here's the difference. Path 1. Constitutive secretion. Think of this like a factory conveyor belt. It's automatic, and it never stops. It is a continuous, unregulated process in which vesicles are constantly transported from the Golgi apparatus to the plasma membrane. It supplies the plasma membrane with new lipids and proteins to replace damaged ones or to support cell growth. Every cell in your body does this 24/7. It does not require a specific signal. Path 2. Regulated secretion. This is the fire station. Unlike the constitutive path, these vesicles, often called secretory granules, accumulate near the cell membrane. They are docked and loaded, but locked in place. It is a controlled process in which vesicles accumulate near the plasma membrane and are released only in response to a specific extracellular signal. This signal is almost always a transient rise in intracellular calcium. When a spike in calcium hits the cell, the floodgates open. This is how neurons fire and how insulin is released. Here, the vesicles are preformed and docked at the membrane, and the release is rapid and stimulus-dependent. Moving to clinical significances of exocytosis. Exocytosis is essential for cells to communicate and secrete important molecules. Its dysfunction can lead to several health issues. Neurons rely on exocytosis to release neurotransmitters. Impaired release can cause disorders like Parkinson's disease and myasthenia gravis. Exocytosis controls hormone release. Problems in this process can lead to diabetes due to insufficient insulin or thyroid disorders. Immune cells use exocytosis to release enzymes and cytokines. Defects may result in immunodeficiency or excessive inflammation. Digestive cells release enzymes via exocytosis. Dysfunction can cause pancreatic insufficiency and malabsorption. Modern therapies use exocytosis pathways for targeted drug delivery, including nanoparticles, liposomes, and antibody-based drugs. And that brings us to the end of this video. Let's take a second to appreciate how much ground we've just covered. We started with the transcellular route, the main highway for drug absorption, and walked through every major mechanism a drug can use to cross a biological membrane: passive diffusion, facilitated diffusion, active transport, and vesicular transport. Each one plays a unique role, and together, they explain how a drug goes from being swallowed, injected, or inhaled to actually reaching its target. Now here's the thing. We've only covered half the story. In Part 3, we're going to tackle the paracellular route, the other major pathway for drug transport. We'll cover exactly how it works, the role of tight junctions, what types of drugs use it, its key features, and why it matters clinically. It's a shorter pathway, but don't underestimate it. It has some fascinating implications for drug design and delivery, so if you haven't subscribed yet, now is the time. You don't want to miss Part 3. If this video helped you, drop a like, Leave a comment with any questions and share it. Until next time, stay curious, keep learning, and I'll see you in part 3.